Imaging, radiopharmaceutical saves lives. I'd like to welcome everyone this morning on behalf of the sponsoring organizations, the Medical Imaging and Technology Alliance, known as MITA, the Society of Nuclear Medicine and Molecular Imaging, and the Council on Radionuclides and Radiopharmaceuticals. I'm Michael Guastella. I'm the Executive Director of CORAR, and particularly excited to be here this morning. I just want to kick things off and introduce our panel and our speakers. We've got Dr. Vaskin Dilsizian, we have Josh Mailman, and we have Terry Wilson. And so, without any further ado, I'm going to turn it over to Dr. Dilsizian so we can start the presentations. Thank you.
Thank you very much. It's really a pleasure to be here. And on behalf of the Society of Nuclear Medicine, where I'm serving as the president, and my faculty positions is at the University of Maryland School of Medicine, where I'm professor of medicine, radiology, and chief of the Division of Nuclear Medicine. So these are fantastic times for our field, and so I'm too enthusiastic. I apologize, but I'm a cardiologist, and I found this field through my research, and I'm head of the Nuclear Medicine Division now because I think this is a fantastic area that's going to be the field that's going to be taking us to a new era in imaging. So first, SNMI stands for Society of Nuclear Medicine and Molecular Imaging, which was founded in 1954. It's the largest international scientific organization dedicated to the field of nuclear medicine and molecular imaging. And what makes it special is that it's a multidisciplinary organization, meaning that it's not just an organization for physicians. It's an organization that includes scientists, radiopharmacists, physicists, and technologists. Next slide, please. So when we think about diagnostic imaging, I think most of us will think about chest x-rays, CAT scans, and it's really used for medical evaluation of body tissue injury. And you look at that with images, you look at the changes, what's normal and abnormal. And we have been using imaging to predict treatment and prognostic outcome of patients. So let me just stop here and say, when we get an image of X-ray, which is an extra beam coming from outside, you're looking at an organ, let's say the kidneys. So you see two kidneys, and you see the size, and you see the anatomy, but what you don't know whether that kidney is functioning normally or abnormally. So I have the same X-ray of two patients with abnormal non-functioning kidney and normal kidney. As far as the X-ray is concerned, it's a kidney. But what we don't know whether they're functioning appropriately or not. Next slide, please. So what is nuclear medicine, therefore? So nuclear medicine involves the use of small amounts of radioactive materials that trace a variety of diseases and looks at the functionality of that organ, such as the kidney. And radio tracers, and I'm not going to do too much physics with you, but they can emit certain particles, alpha, beta, and gamma. And what's shown in the cartoon diagram there is that a gamma ray, for example, travels a long distance. It can go outside of the soft tissue bodies and be able to image by diagnostic camera. So again, the signal, unlike X-ray, where X-ray comes in, nuclear medicine is a signal coming from inside and out where you kind of image that. The alpha and beta particles are short, they travel very short distances, and they're very powerful. They're used for treatment of disease without killing normal cells. So I'm going to go through this with imaging. Next slide, please. So what is a radio tracer? Well, if you think about it, you say, if I want to be looking at a certain enzyme or certain receptor or certain protein in a particular organ, wouldn't it be nice if I can label that protein with some radioactive metal ion, and then that radiopharmaceutical therefore can be injected through the vein and will go to where it's supposed to go based on the ligand, which is the protein, the enzyme, or the receptor, and allow us to image it because you have that radioactive metal ion that's emitting gamma rays. Okay, so that's really the basis of what we do. Next slide, please. So now when we inject this radio tracer, it can go through various organs, brain, heart, lungs, liver, and so what you're now looking at, instead of the anatomy, we're trying to look at the functionality of a particular organ, whether it's upregulated, downregulated, absent, present, it was normal function. Next slide, please. So as I said, the radio tracer is emitted now from inside and out, therefore you put detectors around, in this case, the head, and you can get whole body images, and you can see that image doesn't look like a brain, It just looks like there's a skull there, but what you're looking at is activity in the salivary glands. You see activity in the liver, spleen, and the kidneys. You see like kidney-shaped but intense uptake, and below it is the bladder. So again, not anatomy, but functionality. We're trying to see how things work. Next slide, please. And so molecular imaging, therefore, is a type of imaging of nuclear medicine where you can imagine now we're going to some cellular molecular level of assessing tumor cells, right? Or activity in Parkinson's disease. For example, here you're looking at the activity of whether dopaminergic neurons are acting normally. On the left-hand side, you see this gamma signs intense. That's the normal uptake for patients. And on the right-hand side, you see that the gamma sign, the lower half, is not seen. That would be diagnostic for Parkinson's disease, for example. Again, anatomy. Next slide, please. The anatomy, for example, in the middle, that would be beautiful MRI picture. Fantastic. You look at it and say, well, that's beautiful. But you don't know whether it functions normally or abnormally is where molecular imaging comes in. On the right-hand side is an example of an Alzheimer's patient. Well, again, brain images, as you know, with Alzheimer's can look normal. CT scans, MRI, you don't know why the individual is losing their memory. Yet you can do some type of a plaque imaging with amyloid deposition there on the lower hand. You can see that red-orange site suggests that there's been amyloid deposition in the brain, which is an explanation why this patient, who apparently has a normal brain on anatomy, is not functioning well in daily life. Next slide, please. So, I've just defined to you what radiopharmaceutical is. And as I said, there are diagnostic radiopharmaceuticals. There are the ones that travel the gamma ray, goes outside the body in the image. And then there's therapeutic radiopharmaceuticals, which I will show some examples of. Next slide, please. Now here's a patient, since I'm a cardiologist, I got to show you one cardiac image at least. On the left-hand side, what you're seeing are donuts of the left ventricular myocardium. Now you probably are all used to seeing anatomy. You know, coronary vessels, atherosclerosis, plaque. Well, that's important. But if you, for example, have a 60% narrowing of a coronary artery vessel in two subjects, one of them could have a perfectly normal perfusion, the other one could have abnormal perfusion. So should be angioplasty everybody with 60% lesion? The answer is no, because as long as the physiology of blood flow of the myocardium is preserved, don't touch it. On the other hand, if there's reduction in blood flow, this patient having chest pain, then you will be able to open. You see how anatomy and physiology work together, which is the strength of nuclear medicine. Next slide, please. So here's a patient with a lung nodule. You may be a smoker or a non-smoker, and a lung nodule can be benign. It can be just a calcified granuloma. You may be exposed to some histocytosis or some in travels, and it may be benign. But you see on chest x-ray, you worry. First thing you worry about is cancer. So how shall we manage this patient? Well, in the old days, you either used to monitor it over several months, you keep getting x-rays, see if it's going to increase in size or not. If it does, then you may go ahead and biopsy it. Sometimes the biopsy of a small lesion may be tough, you can miss it, and you may have complications. What molecular imaging does is that I'm going to now introduce a radiotracer that looks at the metabolic activity of that nodule. And if it's hypermetabolic, like it is here, you can see it's like a star. Like, that's not a good thing. If it's low activity or none, then it's probably benign. So now, in this patient, you don't have to wait for serial images to see if it's growing or not. You can go in and perhaps do lobectomy and find the disease early before it spreads. So that's the advantage, again, of molecular imaging. Next slide, please. So here's a patient with colorectal cancer. And on the left-hand side is a CT scan of the liver. You look at that and you say, well, that looks pretty good. On the right-hand side is those hot spots within the liver that tells you, well, hold on for a second. Maybe anatomically you don't see it, but there's actually abnormal activity in the liver, which just has been metastasis to the liver already. Now, what I'm showing you below there is that, as you know, these are FDA-approved agents, and that these imaging modalities have changed management. 37% of patients who had imaging, and then recurrence, again, not knowing the anatomy, physiological, metabolic information provided a third of the patient, they changed the management and treatment because there's such information where you say, liver is okay? Oh no, hold on for a second, liver is not okay. And therefore we should change our approach of managing this patient. Next slide, please. Then we can use this information for the liver involvement before and after treatment. You give chemotherapy. Well, how do you know chemotherapy is working or not? Well, anatomy, the mass may not change with time. It may take a long time for the mass to get smaller. But metabolism is much more rapidly changing back and forth. And so if you treat someone and things are going in the right direction, you're happy to give that news to the patient, even though anatomically things are not changing. Now I'm going to give you this very, very important slide. Next slide, please. Now, most of the time, here's a patient where you have a lot of these lung lesions. Everybody sees that that's pretty bad looking. And then below that is the metabolism. Everybody sees that. It's bad. So now you're going to start chemotherapy in this patient, and most of the time, depending on the chemotherapy, you give 6 cycles of therapy, and that may take 6 months or so. So if you're going to do this blindly or just anatomically, you can see that the patient on the top, after 1 cycle of chemotherapy, metabolism is not any different than where it started from. So which means chemotherapy isn't really working. Now in the past, we wouldn't know that. We just keep doing 6 cycles of therapy, patients getting sick, unnecessary expense, you're losing time or doing something else. And you wouldn't know anything about that until the 6 cycles are completed, and then you hope that anatomically things are shrinking, masses are getting smaller. On the other hand, here's the other patient. Anatomically things look the same after 1 cycle, but metabolism is significantly better. And now if you continue treatment of this patient, you can see 6 cycles later, the anatomy is also getting better. But you could predict that very early on in disease, rather than keep giving chemotherapy unnecessarily, which has, again, implications for cost, side effects, unnecessary patient management, or you could have done something different. Extremely important. This is being used now all the time in most cancer centers. This kind of approach has become part of the guidelines of how to manage patients in cancer. Next slide. Now here's a patient with lymphoma. Lymphoma is a lymph nodes in the abdomen. They all look bad. Things are getting better, and then got better in the abdomen completely, but then guess what? There are new lesions now in the chest. So whole body imaging allows you not only to see where things are getting better, it also tells you whether there's any recurrence of metastasis somewhere else that you may have missed. Next slide. So now you take this concept of precision medicine. We're talking about targeting particular protein, particular cells, particular receptors or enzymes. And what I've shown you so far is that I'm going to use some label to be able to image it. That's all we've done. And to follow the images, improvement or recurrence or progression, right? So the next question you may ask is, if you're able to identify the tumor cells so well, why can't we just kill them there? Instead of giving chemotherapy that goes everywhere, why can't we target the image that you show me, change the radioisotope from a gamma ray emitter to an alpha or beta emitter, which goes to where the tumor is and kills the cells locally with high-powered radiation. Cool. It's a targeted therapy. So that's what we're going to be talking about. You know, what you're showing there is that you can have the tumor identified with a certain antigen, and then you can have now antibody to that antigen labeled with a particular alpha or beta emitter and try to kill that cell. And there are several examples of that already in clinical practice. I'm going to show you one of the exciting ones recently. Next slide, please. Now, these are 8 separate patients. Before and after, before and after, before and after, before and after. To make it simple for you, red is bad, tumor. So before and after, you can see that all of these tumors are completely disappearing. And these are patients with prostate cancer. And prostate cancer cells have prostate-specific membrane antigen, specific for the prostate cancer. Now we can target that with lutetium, which is a beta emitter, that's going to go target where the cancer cells are and kill it locally while maintaining the other normal aspects of the tissues intact. So this is where we are, and this is an amazing area of imaging and therapy that's only going to grow. And you can understand why I'm excited about it, because it's unlike what we've already accustomed to, which is great. CTs, MRIs, ultrasound. But we're talking about a whole new world, right? I mean, if you think about the biological system, we're talking about molecular, cellular, enzymes, receptors. Now, I'm proposing to you that we can actually image these, and not only image, identify at a very early stage of cancer, because we can even treat them. And this is where we are. Next slide, please. So, I'm just going to stop with this excitement, but you can think about where we are now, and then the world, and from several decades and centuries from now, we're going to be talking about genomics, we're going to be talking about proteomics, and you say, well, how do I actually image that? It's one thing to take, blood samples and look at genomics, but don't you actually want to assess this in the whole body with outside imaging? And for that, technology has to get better and better. The resolution of the technology has to get better, and our targets have to be much more specific. But I have no doubt this is the future of imaging, of functional and molecular imaging. Thank you very much for your attention.
Good morning. I get to do the fun part of trying to broadcast in real time that I'm actually talking, which should be a new feed. So welcome. My name is Josh Mailman. I want to talk a little bit about neuroendocrine tumors, which I don't assume how many people who aren't in industry know about neuroendocrine tumors. Show of hands. So a few. That's great. That's outstanding. We'll learn a little bit in where molecular imaging came in for the neuroendocrine tumor, those patients with it. So actually, back one slide. I'm the president of NorCal Carcinet. I've served on the advisory board of the North American Neuroendocrine Tumor Society for the last 4 years. I'm also on the board and treasurer of the Neuroendocrine Research Tumor Foundation. Next slide. I have a few disclosures. No one's seen this presentation before. You guys have seen it in the room. SNMI paid for my travel, and that's about it. Next slide. So first things first, a little bit of who am I. I am a patient. I'm the end user. I'm the person who gets these things. I've had both, as was discussed before, the diagnostics and the therapeutics. But I was diagnosed in 2007 with a pancreatic neuroendocrine tumor, or something that's known as a PNET. We'll talk a little bit about that in a second. My first imaging with molecular imaging was done in 2007. It was an early thing called an Octria scan, which was approved I think either in the late 80s or early 90s. My first gallium-68 scan, and we'll talk a little bit about those images you saw a little right before this presentation, was done in 2008 in Germany. I had subsequent follow-up treatments in Germany with nuclear medicine therapeutics. The term theranostics is used occasionally to talk about the idea of having a diagnostic agent and then swapping out a radionuclide and putting in a therapeutic nuclide. So you're using the same kind of targeting mechanism, but you're changing it from a light bulb to a bomb, and you're able to actually target and really destroy disease. I became involved in advocacy and medical societies a little bit after my first set of images, when I was doing fairly well and asked how could I help. I'm on numerous boards and was the recipient of the SNMI President's Award for my work actually in imaging here. I wrote a little bit about this. If you want to read a history about how it is to be with rare cancer, it was actually published oddly on my birthday. So Living with Rare Cancer, my Dr. Seuss world, it's ASCO post. You can find it readily, pretty easily. Next slide. So a little bit about neuroendocrine tumors. They arise all over the body. It's a rare orphan disease. So what does that mean? There's prevalence is about 7 out of 100,000, or about 21,000, incidence. So there's 21,000 new cases a year of neuroendocrine tumors, but we have some advantage of being able to live fairly long as treatments have gotten better. And so we actually have about 172,000 people in the United States living with it. That still qualifies it under a rare disease because there's under 200,000. it can arise from everywhere. I have a pancreatic neuroendocrine tumor. Many people have things in the GI tract that sometimes referred to as carcinoid. If you look at this kind of really busy slide, it shows where it all can happen, which is lung, stomach, small intestine, appendix, colon, rectum, pancreas. So it can occur everywhere, which is why it's not a really well understood disease or well-diagnosed disease. And it has this, we have many different forms of it. Some of them are very slow growing, actually the majority, which makes it difficult to diagnose and image. We overexpress something called somatostatin receptors. Earlier we heard about prostate-specific membrane antigen. This is a somatostatin receptor, which we all have in our bodies, but these tumors overexpress them and they get used as a targeting agent because we can build something to work with that. So next slide. So how did I find out about advances in nuclear imaging? Because that's really what this whole panel is here to discuss, really advances. And I went to a patient conference in Canada, of all places, in Toronto in 2008, and I was listening with Sandy McEwen, who was the head of this conference on the medical side, and there was a kind of a German nuclear medicine doc who actually, after the first day of the program, gave a slide about this new type of imaging called gallium-68 DOTA something, DOTA talk, DOTA tate. I didn't know because I was a patient, I had no idea. But he put up this slide, which was really interesting to me. And you can see in here was a new type of thing that had sensitivity of 97%, specificity of 92%, accuracy of 96%, compared to the CTs that we were seeing, which were in the 67%, and the other forms of imaging, which were in the 50%. And I sat there, I'm a technologist, my background is, I say that in a nuclear medicine group, it's different. I'm a technologist from Silicon Valley where I create technology, and I look and sit there and go, more data, better. And so I walked up to this gentleman and said, how do I go? How do I get this? Will this show me something different in my disease? And in fact, 2 months later, I was in Germany being scanned for the first time with a gallium-68 DOTA talk or tate, I don't even remember. Next slide. So what is it? How many are, usually are radiologists or trained to read images in the room? There's 2 of them. I'm going to make all of you readers today. You all will be able to do this. Which one can you read better, the one on the left or the right? They're the same patient. Okay, I'm pretty sure you can all tell that the one on the right, you actually can see more disease, and you can see disease in places where you weren't able to see before. And that's actually gallium-68. DOTA, I don't even know which DOTA it is, but compared to what was available before, which was an indium-111 scan. And you can see what happens is you can actually visualize more disease. Greater sensitivity, we saw before. Less radiation for patients. This is what advances can do. When I was scanned in 2007, I had a 10-month-old who is now 12, but I wasn't able to go home because I would have to take care of him because my wife was having a medical procedure on her own. But this new scan could be done in 2 to 4 hours instead of 2 to 3 days. So really interesting changes in how we acquire images and what we can do for patients and patients' ease of life. On top of that, about 1/3, there's several papers that have been in journals talking about the change in management, and about 1/3 of the people who've had this type of scan end up having a change in how they're treated. So the problem was no one was interested in 2008 in this because it was not patentable, it was readily available but really hard to commercialize. Next slide. So really, the road to improvement, approval and reimbursement, I want to thank the society who's here, the Society of Nuclear Medicine who's here as well, because they really worked hard in getting together a group called the Gallium-68 Working Group that met monthly to work on looking at these trials. There was a World Congress of Gallium-68 in 2011, and at that time there was actually 0 places in the United States that were doing Gallium-68. In At the end of that meeting, there were 2 places that had an IND, which is what you do to start studying a drug. But of course, the IND was on 2 separate different drugs, and they weren't measuring the same thing, so we really weren't getting anywhere. By the time the second Diagnostic Congress rolled around in 2013, this was the map of the number of places, because I did a little project to try to track the number of gallium-68s in the world, actually. And by 2015, we had more than a dozen in the United States. In 2016, NetSpot was approved by the FDA with the work of AAA, and today we have over 450 locations. I stopped doing the math because it's too busy to have 450 locations. And in 2017, people started to get billing. I also wrote an article about what that whole process was like. SNMI pathways will have that. So next slide. Patients were really overjoyed about availability. So we started hearing about this because people like me would go talk about it, or other doctors would talk about it, and people were desperate once they started hearing it. People would get different scans. Gallium, turns out, there's the gallium-67 scan, which is used for infection, not anywhere near good for this disease, and people were demanding that their doctors image them with something that wasn't appropriate with them because it had the word gallium in it. So this is how desperate people were for better imaging and better management of their disease. We got lots of comments. It's now part of the NCCN guidelines. It's standard of care. There are hundreds of these going on each and every day around the country. Next slide. So I'm one patient. I'm often brought up to represent patients, and it's hard for me always to do that because I have one singular story. I went on to Facebook, and the group that I run, Norcross Carsonet, is a fairly large support community in Northern California, and I asked for stories. And this is one couple who had the course of their life changed because of a radiopharmaceutical. And their DOTA scan led twice, actually, to both surgery and treatment. This not only pairs and shows you where you might be able to use the paired radionuclide therapy, but it shows you where disease is, and you may be able to have it surgically resected. We'll get to that in a second. Next slide, please. And 2 more stories. People who were told they had no evidence of disease, and then were then found to have evidence of disease, and they could actually go and treat that disease. So we even have last month of someone who was told they had no disease. They had their first scan, and it detected a 2.5 millimeter tumor that they now have been taken care of. Next slide, please. 3 more stories. Several of these are members of my group, and some of them are members of Facebook. You get this idea of people who thought they were actually either cured or stable, and now they found new disease and were able to get treated and go on with their lives. Next slide. So I do this every once in a while in patient conferences. I have to do math while standing up, which is a little hard, but I'll try. I know it's early in the morning and you guys just came down and are probably comfy, but could everyone stand up for a second? So I'm going to make you all neuroconsumer patients. And I'm going to say there's probably 60 in the room. So in a conventional world, I would say the first 2 rows you can sit down. Okay, so in the conventional world, our old imaging was not able to image all these people because it just wasn't sensitive enough. But the people in the second row, stand back up. We image you. We now can manage and track your disease. We can now follow you. Everyone who has a chair, sit down. So for all of you, we've saved a lot of time. We're now going to image you in 1 day. We're going to take 2 hours instead of 2 days out of your life. We probably won't change your disease, but we've made it a lot simpler and easier. We have docs who can see they'll see the fuzzy images on the left, but they'll figure it out, or they'll see the images on the right and figure that out as well. We won't have changed your management. We'll make your life a lot easier. Those people back who are still standing, we've changed your lives. You will now get treated and have actually better outcomes. And we will either have, you will have a higher quality of life, you will have longer progression-free survival, things will be better for you. And 1, 2, 3, 4, 5 of you in the back door, you can leave. We cured you. Or we got close to curing you. We found your disease, we're able to now operate it and take it out of your body. That may not lead to a cure, but it will lead to long periods of having progression-free survival. We'll monitor you every 6 months. But we couldn't do that without the advances in imaging. Next slide. So with that, I thank you. That's my email address. Feel free to contact me anytime you want.
Follow Josh, but thanks for being here today. Thank you to Dr. Disselsian and Josh for sharing your stories. My name is Terry Wilson. I am the chair of the MIDA PET Group, and I am the Senior Director of Patient Access and Healthcare Policy at Blue Earth Diagnostics. And I'm here to close out the presentation, tell you a little bit more about the legislation. But before I start that, I wanted to tell you why I personally am standing here today. I'm a nuclear medicine technologist by training, so I started as a different technologist than Josh, but in nuclear medicine. And I started in nuclear medicine for 2 reasons. The first was that I wanted to make a change in the lives of cancer patients somehow in my career. And the second was that I knew that patients needed answers. And when I saw the first nuclear medicine scan, it was so different than all of the other radiology scans that I'd seen before. I turned to the technologist and I said, how do I get that degree? So it was it very much resonated with me. I remember as a young teenager watching my favorite aunt go through treatment for breast cancer. And back then, the treatment was very harsh. She lost first her independence and then her life to breast cancer while she was going through the treatment. So her quality of life at the end was very poor. As a technologist, I remember talking to the patients. We inject the patient, we spend some time with them, we chat with them, and then they have a little bit of a waiting period, and then they go in the scanner. And I remember the patients who their future plans were resting on the scan that we were about to administer. And so there's so many patients that you touched over the years that you think about and you think back on, wondering how they're doing now, and whether the test that you gave them the information that they needed or wanted. So I've been lucky enough over my career to watch as our technology has improved to the point where we can give those answers that the patients need in much better than, well, you saw Josh's images. Then when I started, it was the image on the left, and today it's the image on the right. So it's incredible. But ultimately, it's about knowing for the patient and for the patient's physicians to make those decisions about how to treat the patient. So a blood test can tell a patient whether cancer is present. A genomic test may tell them what their risk of cancer is, but they don't know where the cancer is. They don't know what the best step for treatment is without knowing where it is and how much disease is present in their body. So having these tests like PET scans for cancer can give the patients, their physicians, and their caregivers those answers that they need to make important decisions about their future care. It can be the difference between a local therapy, whether they're able to go to surgery or have a local radiation therapy, or if they're going to systemic treatment. And that can change their quality of life, whether they're having the systemic treatment or a local treatment. So that brings me to the reason that we're sitting here before you today in this briefing room. All patients should have access to the right testing as requested by their physicians to help them answer the questions that they need. CMS policy today threatens that access. So under the hospital outpatient prospective payment system, CMS deems diagnostic radiopharmaceuticals as supplies instead of drugs. And as supplies, they incorrectly package them into payments within just a very few APCs. This means that the newer precision diagnostic tools that you've seen some of the images of today, you can go to the next slide, sorry. Some of the images that you saw today These are packaged in with products that have been on the market for a long period of time, are generic, and I kind of say jack of all trade because they can be used for many different indications, and they're used in significantly higher proportion of scans than the newer ones are. So the new products are being developed for one individual cancer or one individual disease state, so they are not going to be used in significant proportions of patients. So when they're packaged into these APCs with the generics that are used for mass quantities of patients, then there's no chance to actually change the APC. These drugs are regulated and approved by the FDA as drugs, and under the HOP system, only during the 3-year transitional pass-through period are they considered drugs. So that means that essentially after the 3-year transitional pass-through period ends, the payment in the hospital essentially goes away. Sorry, this is inconsistent with CMS policy in other areas. Under the physician fee schedule, diagnostic radiopharmaceuticals are reimbursed as drugs, regardless of the pass-through period. So we're bringing the legislation forth. Next slide, Dalton. Thank you. We're bringing the legislation forth now after 10 years of trying to change this policy through CMS rulemaking. We're very concerned about these products that are going off pass-through and that have recently gone off pass-through for patient access. There are several states where there are few, and some states where there are 0 PET scanners that are in the non-hospital setting. So in those states, that means that every single PET scanner is in a hospital setting. So when transitional pass-through ends in those states, we're very concerned that the hospitals may stop imaging. That means that those patients would either have to travel significant distances in order to get the scan, or they may not get it at all. So essentially, if those hospitals stop performing the scans and the patients are not getting them, we're rewinding care back to before these products were FDA approved. So we need to make sure that the patients have access to these technologies. They deserve the access to them, and they deserve to know more about their individual disease. Precision medicine cannot exist if you don't know where the cancer is or how much the burden of disease is, or in the neurological space, whether they have a certain disease or disorder or not. Cardiology and nuclear medicine affects all of these areas. So in addition to the concerns about patient access to these products that have been already approved by FDA, this policy negatively affects the innovation in this space. So drug development is risky, it's expensive, it takes a long time to to conduct the trials and bring a product to market and get approval from the FDA. So when investors are looking at products to invest in and they're evaluating them, they're looking not only at the likelihood of FDA approval, but they're also looking at the long-term reimbursement for all of these products. So given the current CMS policy, innovation in diagnostic precision radiopharmaceuticals is at risk. And to fully protect patient access, you also have to protect innovation. Next slide. So the legislation that's introduced by Congressman Peters, Rush, and Holding aims to correct this issue. How does it do that? It creates equity. See the slide here. It creates equity in the payment system by fixing this packaging methodology that I referenced earlier. It is to pay separately for certain precision diagnostic radiopharmaceuticals that are over a cost threshold of $500 per day. These precision diagnostic radiopharmaceuticals, we also have a date threshold of FDA approval after January 1st of 2008. So January 1st of 2008 is when CMS began packaging diagnostic radiopharmaceuticals. So we chose that date threshold to specifically focus in on just the products that have been affected by this policy. And it also prevents some of the older generic products from trying to increase prices so that they can also be included in the legislation. We added a hold harmless provision to the legislation. And the reason for this hold harmless was to eliminate the burden of the patient copay for the products that were included. During the transitional pass-through period, the products that are under pass-through, there is no patient copay responsibility. So the hold harmless language was added to address the patient copay so that this would not be increasing the cost to the patient. And finally, a section of the legislation has been included for budget neutrality. So any increased cost to the system would be spread out throughout the various agencies so that it's not actually increasing the total cost to the HOP system. So our legislation achieves several goals. Protects patient access for the newer precision diagnostic radiopharmaceuticals by ensuring a reimbursement pathway for hospitals. It encourages innovation and ultimately competition in diagnostic radiopharmaceuticals, which is good for the patients. It does not increase the costs for the Medicare beneficiaries, and it maintains CMS packaging for the older generic products, and it's budget neutral. So we appreciate your support of HR 3772. That's a hot off the press number. It came out last night about 7 o'clock. And we're asking that you contact the offices of Congressman Peters, Rush, and Holding to co-sponsor. We look forward to meeting with you in your offices later today and in the near future. And we're happy to take any questions.
But I have a question.
Yeah.
So budget neutral, but in the package material that you gave us, you used the numbers, I think, 10 to 26 million. Is that what's being spread out across all the generics and non-generics?
Yeah, so it was, we have run several CBO simulations, and it appears that the costs could be between 10 to 26 million, but that is spread throughout the HOP system and the various APCs. Yes?
When you were talking about the reclassification of supplies versus drugs, does that affect just Medicare, Medicaid, or does that affect all insurance?
So we're drugs everywhere except the CMS ops system.
Okay.
So it's just correcting the ops designation. Yes?
With any of the imaging that's been discussed here today, were the national labs used at all?
At NIH, for example?
Or the Department of Energy?
Maybe I didn't fully understand the question.
Were any Department of Energy national labs used in any of the radio imaging that's been discussed today? In
They
any part of the
do,
process?
yes. Yes, they
Which,
have.
do you know which lab?
I know thank you, Eleanor.
Hi, I'm with Siemens Healthineers, and our tech business was developed with Oak Ridge Labs in Tennessee, and our business is based there in Knoxville, Tennessee.
Thank you.
Brookhaven National Lab was also involved in development of new nuclear medicine procedures, and some of the other labs provide radioisotopes that are used, including for research purposes for some of the new, especially PET procedures.
And if you want more details, we can follow up with your office. Anyone else?